Hello viewers, welcome back to my channel. Today I'm back with another video, the cell biology zoology session 4. In this the subtopic lysosomes. So what are the lysosomes? Smaller vesicle is called as lysosome. And where they are located? They are located beside the Golgi complex. Golgi complex plays a main role in the formation of the lysosomes. Without Golgi complex, no lysosomes are there. Here, who observed the lysosomes? Firstly, Christian de Duve observed in the electron microscope in 1955. They are tiny, spherical and sac-like structure. Sac means bag-like structure. And dense. Here dense means they are deeply stained and finely granule-like structures measuring about 0.2 to 0.8 microns. Here the microns because they are very minute, and the outer layer made up of particular protein called as lipoprotein. In this, hydrolytic enzymes are present. It is impermeable. The enzymes which are present in this are acidic in nature. Why it is acidic? Because for the digestion of the carbohydrates and etc., what we take the food to digest that, the lysosomes are present. The lysosomes contains enzymes which are acidic in nature. So the enzymes, there are 4 types of enzymes: proteases, lipases, carbohydrases and nucleases. Here what is the role of the protein? Protein converted into the amino acid in the presence of the proteases enzyme. These convert the complex compounds into the simple compounds, or the complex form to the simple form. And then the lipases. What do you mean the lipase? Lip, as the name indicates, these are the lipids or fats. Then it is converted into simpler form. Fat plus glyceraldehyde. The lipids or fat is converted into fat plus glyceraldehyde. And then the carbohydrates. Polysaccharide to monosaccharides. And then the nucleases. Nucleotides to simple form. Polysaccharide to complex form of carbohydrate. Polysaccharide is a complex form. Here the 55% of energy is initiated. It produces the 55% of energy. Then the lysosomes are also called as the cleaning agent because it digests and it cleans. If death takes place, if a living cell, consider you have a living cell and around this the other cells are died. At this situation, if death take place around one cell which is living, the phagocytosis takes place. Here what is the phagocytosis? Dead cells enter into the living cell. The dead cell were received by lysosomes, that is called phagocysis. The dead cell were received by the lysosomes to protect the tissue from damage. If one dead cell is present, it also affects the other one. So a cell receives the dead cells through the lysosomes to protect the tissue from damage. Phagocytosis is very important to protect cell from further damaging. Then came to the apoptosis or PCD, programmed cell death. For the cell death we can predict. For us or for the animals, no one can predict when he dies. No one can predict his death time. but for cell we can predict it. So the RBC life is 120 days only. As this, we can also predict the date of the death of a cell. For the cell death, we can predict when the cell will die. So prediction of death of the cell is called apoptosis. During releasing of enzymes, lysosomes get permeable. Enzymes came out to hydrolyze the dead cells, which enters the cell during phagocytosis. After entering the cytoplasm, the lysosomes enzymes become soluble. Aging is also shown by lysosomes. For example, we human after 35 years or the we women after 35 years will look like a age because of the lysosomes. And then the mechanism, second mechanism, mitochondria are also have death. If mitochondria dies, there is no energy production. Mitochondria provides the energy for the activity. Mitochondria also have death. During this time, cells also get itself damaged. It is a powerhouse of the cell. If mitochondria dies, then the cell doesn't function anything. So mitochondria is very important. Mitochondria also have death during this time. Cell also gets itself damaged. This mechanism is called autorhagy. 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 Suicidal bag. Because of lysosomes, only cell get disrupted. So it is called suicidal bag. Here the lysosomes is converted into or goes into the cytoplasm and the mitochondria dies. Helps in cleaning up. And then so mitochondria also destructed cleaning, so it is called as suicidal bag. It is destructed or it is cleaned, so it kills the powerhouse of the cell, so it is called as the suicidal bag because a cell itself is damaging. And then the polymorphism. What is the polymorphism? Like for example, one person does two or more than jobs. This is like a polymorphism. A general example to understand in a simpler way what is the polymorphism. Here the lysosomes are different types and different in different cells. Lysosome is polymorphic in nature. Primary and secondary lysosomes are present. Primary lysosomes, Golgi complex responsible for secretory vesicles, and one of vesicle become lysosomes and it is called primary lysosomes. Here the vesicles we know as the vesicles carries synthesized protein. So one of the vesicle is smaller than as compared to the other, so that is called as the lysosome. So the enzymes are made up of proteins as we know before, and then the Golgi complex to lysosome. Then the secondary lysosome or the foreign bodies, phagosomes means the foreign bodies, enters the dead cells. Along with the dead cell, foreign bodies also enters and dead cell enters the living cell. The primary lysosome attached particular bacteria. The primary lysosomes attached to the particular bacteria. Here the fusion takes place and form secondary lysosome is also called mature lysosome. Secondary lysosome is important for releasing the enzymes. So this is regarding to the whole topic of the lysosome. I wish you guys can understand it. Thank you. Please do like, share and subscribe to my channel. Thank you for your support. Thank you.